Hello, AP Bio. Today, I more than likely have jury duty, and that's why you are watching this video. But today, we start our journey into cellular respiration. And with cellular respiration, keep in mind this is an order of a ton of different steps, and this is a protein and enzyme driven reaction. We need to keep these concepts in mind when we talk about it. And we need to first start with, if my computer wants to work, there you go. We gotta talk about the mitochondria because the mitochondria is in charge of cellular respiration. And while we right now don't know a ton of about cellular respiration, we do know as a class this is the process of which we grab food and we turn it into energy in the form of ATP. When we look at the mitochondria, we have various different parts. First things first, this is a double membrane structure similar to our cell membrane, a bilayer. It has a smooth outer membrane. This assists in the mitochondria making its way through the cell. Additionally, it has a inner membrane. This inner membrane is highly folded and is called the cristae. We look back on our journey through cells and specifically cell size when we talk about surface area to volume ratio. We said prior that surface area to volume ratios, we want it as high as possible because when that number is high, we have an efficient cell. This is no different. We want to increase the surface area and volume ratio of the mitochondria through folding the inner membrane. And we will see this is incredibly beneficial for the process of cellular respiration. Additionally, we have an intermembrane space. Think of this like a fluid filled space between the membranes. This is filler, right? The mitochondria, if it lacked structure, could, in the theoretical, collapse in on itself without that set structure. Think of like a house, right? You don't just have four walls and call it good. You have a bunch of supporting walls. You have beams. You have things that keep the house afloat. So does the mitochondria. The next part we talk about is the matrix. This is the inner fluid filled space. The reasoning why we talk about the matrix and the cristae, or the inner membrane, is because each of these do a separate job. We will learn the cristae does a process called chemiosmosis and the electron transport chain. And the matrix is in charge of what's called the Krebs cycle. We already kind of know from our previous unit that the mitochondria has its own DNA, it has its own ribosomes, it has its own enzymes, proteins, and these enzymes and proteins can be found anywhere. It could be found in the matrix, it could be found in the membrane, but the mitochondria, as we believe as scientists, was once its own organism. It was its own prokaryote that became part of a larger cell. Now, a question we need to ask ourselves: what cells would have a lot of mitochondria? Insert Ed puzzle question now. Well, we look what cells would need the most amount of energy. We look at humans as the example. Well, for us, our brain cells, our muscle cells, our heart cells are highly needed cells that need a ton of energy. The cells will adapt and make more mitochondria or accept more mitochondria in comparison to some of the less energy needed cells, such as skin cells. So, cellular respiration overview, we look at it as a catabolic pathway. So, pathway is a series of different steps. Catabolic means to break down. If we remember, that prefix cata means down. So, we are not building anything, we are breaking apart. In this scenario, in cellular respiration, we look at that molecule glucose that we have heard about time in and time out before. So glucose is going to be broken down over a series of reactions. We will go in depth regarding these reactions, but you're not expected to remember exact numbers. So we will talk about that in the electron transport chain, that there's 36 ATP that are made. You are not expected to remember 36 exactly. So, items like that are unneeded to remember. But conceptually, we do need to know what is going on. Additionally, through the process of breaking glucose down, we're going to result in a ton of electrons. If we remember back from cellular energy, electrons are the main source of energy in an atom. These electrons are going to be shuttled or moved around by a molecule called NADH, and that's going to be transported to the electron transport chain. So we're looking at our steps. We will talk about step one, which is glycolysis. That is the focus for today. Then tomorrow we will look at the Krebs cycle. Then we will look at the electron transport chain and oxidative phosphorylation. But we look at these terms first. Glycolysis, the word lysis means to break. Glyco usually refers to sugar or fat. In this case, we are looking at sugar. Glycolysis, like we just said, is sugar splitting. We are breaking sugar. This happens not in the mitochondria. This happens in the cytoplasm. If we remember back from Unit 2, the cytoplasm is just a filling in the cell. It's like jelly-like substance. Sorry. This process, we will learn that oxygen is critical in deciding which pathway we go down when we're trying to make energy. This process does not care whether we have oxygen or not. And in fact, this is believed to be a ancient trait in which bacteria that first originated on Earth did not have sufficient amounts of oxygen, so had to result in breaking apart glucose to derive energy. So we look at this diagram. We are looking at our glucose. Glucose is a 6-carbon sugar that is going to be broken down into 2 pyruvates. Be familiar with that term, pyruvate. Pyruvate is a 3-carbon sugar. So we look, and mathematically speaking, 6 divided by 2 is 3. So we result in 2 pyruvate, each with 3 carbons. However, there's an additional step we'll talk about in just a second. This whole process of glycolysis results in 2 ATP and 2 NADH. Additionally, it results in the creation of water. So why do we care about NADH and ATP? Well, number one, ATP is the energy of the cell, right? We want energy. But NADH is critical to the next 2 processes that we talk about, either the process of fermentation or the process of cellular respiration. We call it net gain. The reasoning for it is because for glycolysis to happen, think about when you're breaking something. You don't just break it with your mind, right? You have to put energy into breaking an object. The cell is no different. It has to put energy into breaking glucose. Specifically, it spends 2 ATP and results in 4 new ATP. We do 4 minus 2. That gives us 2 net ATP out of it. So keep this whole process in mind. Glycolysis is always going to happen, regardless of oxygen or no oxygen. When we talk about these 2 paths, we either talk about aerobic and anaerobic. For those that are in sports or have played sports before, you have done aerobic exercises and anaerobic exercises. Aerobic, we look at the prefix AERO or aero. That means air. It is a process that requires air or oxygen. Anaerobic, AN, usually means without. AERO, oxygen or air, without oxygen. So where we talk about the aerobic pathway, no, we're talking about oxygen. Anaerobic, without oxygen. More specifically, when we talk about glycolysis, we have 2 phases. Energy investment is phase 1. This is endergonic. Think about it, what it means to invest. You're putting something into a process. In this case, we are putting energy and we are investing it. We're putting energy into the reaction. This costs us 2 ATP that result in 2 ADP. From there, we get two molecules. Both of them are what is called G3P or glycerylate 3-phosphate. Keep in mind with these phosphates and the importance of them, because if you remember, ATP is made of three phosphates. Breaking phosphates usually results in energy. So this is energy investment. We haven't gotten pyruvate yet, but we've broken glucose down. Then we look at the next phase, energy payoff. This is exergonic. We are releasing energy. We have two handy molecules that are going to come in handy during this process, NAD plus and ADP. Because what's going to happen is NAD plus is going to pick up hydrogen and electrons from this molecule. Additionally, ADP is going to pick up the phosphates, resulting in 2 per G3P. If we remember, in energy investment, we result in 2 G3Ps, right? We get 1 NADH, 1 NAD, and 2 ATP per G3P. So we are going to result in a total of 4 ATP, 1 from the first, 2 from the first, 2 from the second, and 2 NAD, 1 from the first, 1 from the second. In this case, we say NAD plus is oxidized by giving the hydrogen to it. Additionally, we use a molecule called PEP, which donates the phosphate to ATP. This is called substrate level phosphorylation. This is an enzyme-driven process, right? We looked at it, we said that's why we covered enzymes. This is enzyme-driven. So an enzyme is grabbing phosphate and attaching it to ADP. We look at glycolysis and we talked about two pathways, fermentation and respiration. Fermentation is anaerobic. It's not going to use oxygen. Respiration is aerobic. It requires oxygen because we learn oxygen is the final electron acceptor. Today we are only going to look at fermentation. Respiration will be all of the following steps Tuesday and Wednesday. So some stuff about fermentation. This keeps glycolysis going. So essentially we just keep breaking sugars over and over again by donating the hydrogen given to NAD. This occurs in the cytosol, aka cytoplasm, and we either result in a product called ethanol or a product called lactate. Additionally, we get 2 ATP per glucose molecule, right? This only happens whenever we don't have oxygen and is deemed inefficient. Their respiration is so much more productive than fermentation. The only perk of fermentation is we don't need oxygen at all for this to happen. So lactic acid fermentation, we grab pyruvate, more specifically, we grab glucose, break it down into pyruvate, as we see in the diagram. We don't have any oxygen to accept hydrogen from NAD. So then it is recycled into the molecule lactate. Lactate is usually used to make cheese, yogurt, acetone, or methanol. Scientists used to believe that lactate was what caused muscle fatigue. Like if you grabbed a ball, like a tennis ball, and you kept squeezing it over and over and over again, you start to feel a pain in your hand. They used to believe that was lactic acid buildup. However, scientists have now found that that isn't the case. Whenever the process of lactic acid fermentation can be not necessarily reversed, but the tides can change. If the cell is going through lactic acid fermentation and finds itself with oxygen again, all of that lactate that was produced can be turned back into pyruvate through the liver. However, that is a process we do not cover in AP biology. You would need to take biochemistry or endocrinology. Alcoholic fermentation is the other case. Same thing. Glucose gets broken down. We end up with pyruvate. Then we release carbon dioxide and we make what's called acetyl acetaldehyde, which results in the production of what is called ethanol. Ethanol sounds familiar because we're talking about gasoline, but ethanol is also utilized in brewing, winemaking, and baking. This is your yeast, this is your bacteria. Over time, the more ethanol that is produced, it begins to kill the yeast and bacteria within it. So we look at when you make alcohol, usually add yeast. Yeast produce the ethanol, and over time, all that yeast dies away. So those are our two types of fermentation. These are the products of a anaerobic respiration, anaerobic process without oxygen. Tomorrow, we talk about what happens when there is oxygen. You guys, take care. Get these down in your notes. Repeat that puzzle all you want. You guys have a beautiful rest of your day and make good decisions. Bye bye.